Hello. Welcome to the next in the series of review clips designed to assist you in preparing for your Biology 3201 public examination. In this lesson, we'll take a look at genetics, specifically Mendelian genetics. Before we begin, there are a number of terms that you need to be familiar with in order to understand the lesson as we move forward. The first of these is the word gene. Gene is simply a unit of heredity. Genetics, this being the science of heredity. Genes. This term, genetics, was coined at the beginning of the 20th century, and it replaced previous studies of generation, inheritance, heredity. In the 1920s, classic genetics was often referred to as Mendelianism, and was a relatively new at the time, and controversial, I might add, area of biology. But by the 1950s, genetics had become recognized as a unifying principle at the core of the life sciences. So why do we even bother studying genetics? Well, the first reason that understanding genetic processes is fundamental to comprehension of life itself. Genetic function, cellular function, external appearance, linkage between generations are all tied into genetics. Indeed, our modern society depends on genetics. Breeding programs led to crops, livestock. Biotechnology programs produce drugs. Forensic science certainly relies very heavily on DNA identification. And we can't ignore the fact that genetics is indeed a key component in medicine. It's estimated that at least 30% of pediatric hospital admissions have a direct genetic component. Before Mendel, and we'll talk much more about him a little later, plant and animal program were certainly very key parts of human history. Between 8,000 and 10,000 BC, we domesticated horses, camels, oxen, and dogs. We knew that primitive cereal plants, maize, wheat, rice, they were developed about 5,000 BC. And while humans did manipulate heredity, did manipulate species, we weren't fully aware of how it was that this happened. Before Mendel, the dominant idea was that of blending theory, that traits would blend like liquids. Heritable features, those passed down, would become irreversible, inseparable. To support this, individuals would eventually blend to a uniform appearance between individuals, but traits would not be able to reappear once blended. The blending theory, because of these inadequacies, was replaced by the particulate theory in the 1860s. With the particulate theory, the belief was that parents transmit their offspring discrete inheritable factors that we now call genes that remain as separate factors from one generation to the next. Mendel demonstrated this theory by breeding pea plants with specific variants, types of trait contributing towards a character, something they could see. Now, the idea of genetics was certainly something that's been around for a while. Back in the age of the golden age of Greek culture, attention was given to the study of reproduction, the study of heredity. The great philosopher Hippocrates, he argued that both parents contributed traits to the offspring, while Aristotle believed that the male provided the form for the embryo, while the female only worked as an incubator. Hippocrates recognized that the male contribution to a child's heredity was carried in the sperm. Aristotle thought that all inheritance came from the father, and female children were the product of interferences from the mother's blood. And Leeuwenhoek actually saw sperm underneath the microscope. And in so looking, he imagined that he saw what he termed to be a homunculus. What he actually saw was a nucleus in the sperm cell. He thought it actually looked like a little man. And Leeuwenhoek agreed with Aristotle's idea that children were born in the father's sperm. Between 1600 and 1850, the dominant idea was that of preformation, that sex cells contained miniature adult homunculus, a little man. Another theory that arose regarding trait transmission was a theory of pangenesis. And Darwin himself argued the idea of gemmules, little pieces representing each body part, was gathered by the blood in the semen. A piece of an arm, a piece of a leg, a piece of a spleen, all gathered up there and transmitted in the semen to give rise upon fertilization. There is also the theory of epigenesis, where an organism is derived from substances present in the egg, which then must first be assembled and must differentiate during embryonic development. But by the 1800s, it became generally accepted that offspring were a combination of traits from mom and from dad. Inheritance was considered to be due to blending, because many traits that we could see were due to a combination of traits from moms and dads. So when Mendel began his work, the idea of blending inheritance was certainly very popular. This idea was put forward to account for the fact that offspring show characteristics similar to both parents. But who was Gregor Mendel? Well, Gregor Mendel, he didn't have an easy time growing up. He had a number of hardships, financial and health, but he did do well in school. And he actually went to the Olmütz Philosophical Institute in Austria. In 1843, he entered a monastery, the Augustinian monastery, and he was sent to the University of Vienna after he failed his teacher's exam. For a while, he taught at Brunn Modern School. But around 1857, which is the reason why we know him today, he began breeding pea plants in the Abbey garden. Now, there's a few terms we need to understand as it pertains to Mendelian's experiments as well. First of these is the parental or pea generation. That is the original generation of pea plants. That's the original parent generation. The F1 generation, those are the offspring of the parents, parental generation. The F2 would be the offspring from the F1s. True breeding always produces plants with the same traits as the original parental generation. And the experiments that Mendel derived two laws, the law of segregation and the law of independent assortment, which we'll discuss in a later lesson. So to try and diagram this out, we have our original parental generation, P1. P1 produces offspring. This would be the F1 generation. The F1s, when crossed with each other, produce the F2 generation. So what did Mendel do? Well, firstly, he looked at 7 yes or no traits. Height of plants, tall or short. Flower color, purple or white. Seed color, by way of example, yellow or green. He didn't look at quantitative traits such as weight. All traits were simply yes or no. He spent several years getting pure breeding plants for these 7 traits. In other words, a yellow-seeded plant is only going to have yellow-seeded offspring. And what Mendel hypothesized, but there are alternative forms of genes. Now, we didn't use that term. Alternative form of the units that determine heredity. Traits came in one of 2 basic forms. He also hypothesized that one form will be dominant over the other. And here we see a listing of Mendel's 7 traits. Dominant, recessive. Seed shape, round or wrinkled. Color, yellow or green. Flower color, purple or white. Flower position, on the side, axial, or terminal, on the tips. The pod color, green or yellow. Pod shape, inflated or constricted. And height, tall or short. So why the garden pea? Why would you select this symbol plant? Well, there are a number of different reasons for this. Firstly, many hybrids were previously produced. He knew that certain traits segregated the offspring. He already knew this. There are certain ready-to-go traits they could work with. He also knew that there were a large number of true breeding varieties. He studied 32 and he chose 7 that would breed true for him. Pea plants are small, they're easy to grow, and they have short generation times. In other words, you can get a lot of data in a relatively short time frame. And because the sexual organs are enclosed in flowers, they're easy to contain, easy to control the transmission of gametes back and forth between the 2. All these things added to the pea plant being an ideal plant for him to use in his studies. Again, just refresh your memory, character, those are heritable features. Those are a specific property of an organism. For example, flower color is a character. A trait, on the other hand, are different forms of that character. A flower being white, for example. And each pair of Mendel's plant lines demonstrated a character difference. Flowers, white or purple. Height, short or tall. Seed shape, round or wrinkled. So what did he do? Well, his experiments involved genetic crosses. In other words, he mated plants bearing purple flowers and one having white flowers. This created a hybridization where 2 pure lines came together. Remember, the parents are the P generation. The first set of offspring are the F1, first filial. And when you breed the F1, you create the F2, or second filial. And Mendel followed his experiments across 3 generations, P, F1, and F2. So what he did was, firstly, he would take the stamens off of a purple flower to ensure it wasn't self-breeding, and he controlled what sperm was breeding with what. He then would transfer pollen from the white flower to the female organ, the purple. Of course, the pea plant would breed after having pollen exposed. You produce seeds. These mature in the pod. He would take those seeds then and plant them, and looked at the offspring. In this case, it was all purple flowers. Purple being dominant. So, his experiments were all done in 3 stages. First, the plants were self-fertilized. This ensured that the traits were true. Tall plants always producing tall, purple flowers always producing purple, and so on. Second, he crossed plants with alternate forms of trait, removing the male part of the flower from a tall plant, for example, fertilize it with pollen from a short plant. He would then do the reverse as well. As you can see here, pollen will transfer white to purple, all purple flowers in the F1 generation. Third, these hybrid plants would self-fertilize for several generations. And what Mendel did was he followed traits for several generations, and he quantitated those results. He took very meticulous notes to actually give credence to his ideas, document his ideas, document his evidence, his data. And from Mendel's experiment, he deduced that an organism has 2 genes, alleles, for each inherited characteristic. 1 characteristic comes from each parent. This was his principle of segregation, and described the inheritance of a single characteristic. So as F is P generation, purple crossed with white. This yielded all purple flowers. Now we'll talk about this more a little later, but when he crossed the F1s, he created a ratio. He got back the white. It gave you a 3-to-1 ratio. Now, for Mendel, it didn't matter if he crossed a purple male and white or purple female and white. The pattern was true. In the F1 generation, all the flowers are purple. There was no blending. And even in the F2, there was no blending. It was either purple or white. The convention that Mendel started using was the dominant allele, the dominant form, is represented by a capital P, or in this case, a purple flower. PP, 2 big P's, is said to be homozygous dominant. Both the same, both for the dominant form, in this case, purple. Little p, the lowercase letter, represents the recessive allele. In this case, it's white. Big P, little p is heterozygous, 1 of each. 2 little p's is homozygous recessive. Homozygous recessive, little p, would be a true breeding white flower. It can only pass on little p. So again, his first cross between a true breeding purple plant and a true breeding white. The genotype of each parent, each plant. Now, genotype is a type of gene. Because they are true, a true breeding purple plant would be big P, big P. A true breeding white plant would be little p, little p. In terms of the kind of gametes that each can produce, the big P can only produce big P. It's true. The true breeding white plant is little p, so it can only produce little p gametes. As for the genotype of their offspring, if one parent passes on a big P and the other parent passes on a little p, the genotype of the offspring is going to be heterozygous. And as for the phenotype of the offspring, they are all going to be, in this case, purple, because purple is dominant. Next, he allowed the F1 to self-fertilize to form the F2. In other words, they cross with each other. So he had a big P times little p. And what did you get? One tool that can be used to determine what you get is something called a Punnett square. A Punnett square is a tool, a technique to solve genetics problems. And it was named after an English geneticist, Reginald Crundle Punnett, who first started using this method. So, getting back to pea flower, the flower has reproductive structures, male and female. transfer pollen back and forth between the two. In this first generation, all purple colored plants. So Mendel's cross of plants for pea color is as you see here. This tool of using a Punnett square is simply a 2x2 table. That's all it is. In the case of Mendel's first initial crosses. All you do is you isolate the possible gametes and pair them up. In Mendel's first cross, pure white crossed with pure purple, the only gametes that can be produced is a big P or a big P. Every plant has two, so you put them in. 1, 2. You do the same for the other parent. Again, it can produce two gametes. 1, 2. And you then simply line them up. Big P, little p, big P, little p. Big P, little p, big P, little p, big P, little p. All of the progeny, all of the offspring, are heterozygous. To complete as F2, you do the same thing, except the difference now is because each plant has a little p that it can pass on, the combinations are a little different. Big P, big P, big P, little p, big P, little p, but the recessive form reappears when the two little p's combine in the F2 generation. So after the first offspring, which was heterozygous, self-fertilized, you got 3 purple, 1 white. This 3-to-1 ratio is characteristic for a single trait or monohybrid cross between heterozygous individuals. We now know that these traits are carried on chromosomes. We pass on chromosomes. So if you have a homozygous parent, which only contains big A, it will pass on big A or big A. It'll pass on 2, one of each, but if it only has 2 big A's, both of them must be big A. If the parent is heterozygous, big A, little a, its gametes will be either big A or little a. Again, heterozygous individuals can pass on one or the other. So in the case of Mendel's plants, big P could only pass on big P. Little p could only pass on little p. In the F2 generation, when the big P's and little p's combined, you created heterozygous individuals. Whether that was a purple sperm with a white egg or a white sperm with a purple egg. The net result was the offspring were all heterozygous. They had the purple phenotype, but their genotype was heterozygous. When you took the gametes from the F1 plants, the net result would be 3 purple, 1 white. Diagram of the Punnett square, that ratio holds true. 1 white, 3 purple. Again, getting that characteristic 3-to-1 ratio. We know that when you breed true, all heterozygous, but you do get this offspring being created, which is white. The white would reappear. Just to recap, remember Mendel worked with 7 traits. They were all either yes or no, smooth or wrinkled, yellow or green, inflated or constricted, green or yellow, purple versus white, at the leaf junctions or at the tips, axial or terminal flowers, and tall versus short. So just to summarize, Mendel's terms: unit factors are the 2 factors that Mendel said affect the expression of traits in an organism. Each organism has 2 factors for each trait. We now call these genes. The unit theory of inheritance for Mendel, his theory, where he believed that unit factors, we now call them genes, control the expression of traits in an offspring. Mendel's dominant factor, that was always expressed in an organism, if present. The recessive factor, the factor that sometimes was expressed in an organism. Allele, the form of a gene can be, it can be dominant or recessive, but not both. The F1 generation, or first filial generation, the first set of offspring produced by the parents. The F2 generation, or second filial generation, is the second set of offspring created from the offspring of the parents. These offspring are the same thing as grandchildren. Pure breed or strain, a factor refers to organisms having either 2 dominant factors or 2 recessive factors. For example, pure tall has 2 dominant factors for tallness. One from mom, one from dad. Pure short, on the other hand, 2 recessive factors for tallness. One from mom, also one from dad. A hybrid is a term meaning an organism has 1 dominant and 1 recessive factor. We also term this to be heterozygous. Homozygous, term meaning an organism has the same alleles for a gene, both are recessive or both dominant. It's the same thing as pure breed. Heterozygous, different alleles for a gene. 1 dominant form, 1 recessive form. It's the same as a hybrid. Genotype, this is the genetic makeup of an organism. It's the particular combination of alleles. For example, homozygous dominant, big T, big T. Homozygous recessive, for straight hair. Phenotype, that's what you physically see. It's what the observable characteristic. Tall, curly hair, you can see those characters, you can see those traits. Punnett square is a chart that helps determine the possible probable outcomes of a cross. You can see the diagram here, which illustrates. And a cross, this is, simply put, a female has been mated with a male. In the past public examinations, questions have been posed on this topic. This one here, what is an alternate form of a gene? An alternate form. An alternate form of a gene would be an allele. This question from a past public examination reads, which is a study of variation and inheritance in organisms? Evolution is posed by Darwin. It talks about how organisms change over time. Physiology is their physical makeup, how the body structures interact. Zoology is simply the study of animals. The correct answer is B, genetics. And finally, what are Mendel's unit characters known as today? Mendel had no reference chromosomes. Genotype is a particular combination of genes. Phenotype is a physical result, what you see. The correct answer is B, genes. Thank you for your attention. I hope that you found this learning clip helpful.